Biochemistry. Today we are going to study glycolysis, but before studying glycolysis, let us discuss something about the carbohydrate digestion. What type of carbohydrates do we eat in our diet? Mostly starch. Mostly starch. Kind of 90% of the carbohydrate is starch. And then there are some disaccharides as well, like sucrose, like lactose, like maltose, like isomaltose. We take in all that starch and the mouth, chew down, and carbohydrates digestion starts in the mouth. The carbohydrates digestion starts in the mouth by salivary amylase, and the starch is broken down into dextrins, kind of 8 to 10 glucose molecules. Nothing happens in the stomach, and when the carbohydrate enter in the duodenum, they are acted upon by the enzymes which are salivary, which are the enzymes of the pancreas, like pancreatic amylase, and that pancreatic amylase digests the starch further and convert that starch into disaccharides. And then finally, in the enzymes from the intestinal brush border, which are disaccharidases, those disaccharidases hydrolyze the disaccharides into monosaccharides. Let's see how. You see that sucrose, chini, sucrose, sugar and chini in Urdu. Sucrose is hydrolyzed to glucose and fructose. Maltose is hydrolyzed to two glucoses. Lactose, present in milk and milk products, converts glucose and galactose. Isomaltose, that is hydrolyzed to glucose and glucose. And you see that mostly we have glucose. And in addition to glucose, we have fructose and galactose. And this glucose, from our previous videos, we know that glucose is oxidized to pyruvate. And that oxidation of glucose to pyruvate is called glycolysis. This is called glycolysis. And interestingly, fructose also makes pyruvate. We will study the metabolism of fructose here. And galactose is also hydrolyzed to, oxidized to, pyruvate. Interesting. Right. So we are going to study in this chapter the glycolysis, the fructose metabolism, and galactose metabolism. But before moving forward, let us see how the glucose is transported across the cells. How the glucose is transported across the cells. And as we know, glucose is a big molecule, so it needs carrier protein. Carrier protein. And that carrier protein, which transfers glucose across a cell, is called glucose transporter. And we give it a very beautiful name, and that is called GLUT. GLUT. And there are four types of GLUTs: GLUT 1, GLUT 2, GLUT 3, and GLUT 4. Right? GLUT 1 is present in most tissues. It is present in most tissues. Right? And important that you have to remember all the time, that GLUT 1 is also present in brain and red blood cells. Okay? GLUT 2 is present in, most important, in liver and in islets of Langerhans. And you know where are islets of Langerhans present? They are present in pancreas. And GLUT 3 is present in most tissues. GLUT 3 is present in most tissues. GLUT 3 is present in most tissues. And GLUT 4 is present in muscles and adipose tissue. Each GLUTs have specific km value They have specific Km value, and that is very much significant. And that is kind of scientific thing, and scientific things are also often boring. But let me make a generalization of that scientific thing. And what is that generalization? If Km value of anything, mostly the protein, the enzyme, if the Km value is low, it means the protein has high affinity for the substrate, right? And if Km value is high, the protein has low affinity for the substrate. And here the substrate is glucose and the protein is GLUT. Let me make it clear. You see here, the proteins that have low Km value, they will work all the time because they have high affinity for the substrate. They will take up the glucose and shift the glucose into the cells. Whether there is lot of glucose in the blood or there is small glucose in the blood, they will shift the glucose. But they will be slow in action. Well, the proteins or the enzymes which have high Km value, they remain there. They are kind of nerds. They don't want to work. They really don't want to work. They don't work until they pile up a lot of work. And when there is a lot of work piled up, they will start working and they will finish the work in instants. Alright? They will finish the work in instants. And you see that the highest Km value in this condition is of GLUT2. Its Km value is kind of 15 millimole. It will not work if there is small amount of glucose in the blood. But if there is large amount of glucose in the blood, it will shift all the glucose from the blood into the liver in instants. Right? While the GLUT1 and GLUT3, they have very low Km value, that is just 1 millimole. And as they have just 1 millimole Km value, they work all the time. They transport the glucose from the blood to the cells all the time. And GLUT4, which is present in muscles and adipose tissue, they have 5 millimole of Km value. It means they are also under the influence of insulin. So GLUT2 and GLUT4, remember, GLUT2 and GLUT4 are under the influence of insulin, while GLUT1 and GLUT3 are independent of the action of insulin. Whether insulin is there in the blood or it is not there in the blood, GLUT1 and GLUT3 will work. And that is the reason that GLUT1 is present in the brain and red blood cells. And brain and red blood cells need constant supply of glucose. They will die if the glucose is not present in them, if the glucose is not given to them. Right? So understand this. GLUT1 and GLUT3, they are present in brain and red blood cells to ensure the constant supply of glucose to the brain and red blood cells. While GLUT2 is present in the liver and it works only when there is lot amount of glucose in the blood. And GLUT4 is present in the muscle and adipose tissue. They work if there is insulin in them. But how this works? What is the action of insulin? How insulin affects this all? Let us see that. You see here, this is a cell. And the GLUTs are playing around in the cells. There are just 1-2 GLUTs present all the time. These are the GLUTs, they are present all the time. But the remaining GLUTs are there enjoying in the cell. And insulin comes. When insulin comes, these GLUTs start appearing at the surface. And once they start appearing at the surface, the glucose gets in. And as soon as insulin disappears, these GLUTs move inside again, so that the entry of glucose will decrease. Is it clear? Right? These are the doors. GLUTs are the doors. Right? They will open when the insulin is there, and they will be closed when the insulin is not there. I hope this is clear. Right? The GLUTs are the gates which transport glucose from outside the cell into the cells, or from inside of the cell to the out of the cell. okay, and they are under the influence of insulin. All right, now we formally go to the glycolysis.